Good afternoon and welcome to part 2 of the Provost lecture mini-series dedicated to the COVID-19 pandemic. My name is Fotis Sotiropoulos and I'm the interim Provost and Senior Vice President for Academic Affairs at Stony Brook University. Vaccine development normally takes many years. Yet in the past 11 months, we have been witnessing what could be arguably characterized as one of the most impressive scientific undertakings in the history of humankind. It was January 11, 2020, when Chinese authorities first shared the full sequence of the novel coronavirus genome. And on that day, the fast and furious race to develop, test, and get approval for the vaccine was on all around the globe. And just this week, less than a year ago, the United Kingdom became the first Western nation to authorize a COVID-19 vaccine and announce large-scale vaccinations of its citizens to begin in days, and with the United States and many other countries very soon to follow. This has to be one of the most striking scientific accomplishments, attesting to the boundless potential of human ingenuity and the awesome power of the convergence of biology with biotechnology and information technology. Today, we have put together for you a mini symposium with 3 talks that will take us through the basic biology of the immune response to foreign invaders, the current status of clinical trials of various vaccine candidates, and the efforts to develop passive immunity. Our first speaker today is Dr. Ken Kaushansky, the Senior Vice President for Health Sciences at Stony Brook University and the Dean of the School of Medicine. Dr. Kaushansky is a world-renowned hematologist, physician, scientist, and teacher whose transformative contributions to understanding hematopoiesis have been recognized by his election in the Institute of Medicine and the National Academy of Sciences. His research has led to several significant discoveries, for which he received the Dameshek Award from the American Society of Hematology, the Outstanding Investigator Award from the American Society for Medical Research, and the Ernest Butler Award from the American Society of Hematology. Dr. Kaushansky is an inventor on 6 issued US patents that have been licensed or assigned to 2 companies. He has published over 140 peer-reviewed papers and over 115 invited papers, editorials, or textbook chapters. He has served as editor-in-chief of Blood and on the editorial board of The Hematologist. He currently serves as lead editor of Williams Hematology, Hemostasis, and Thrombosis. Dr. Kaushansky is a fellow of the American College of Physicians, member of American Academy of Arts and Sciences, and a master American College of Physicians. Without any further delay, it is my pleasure to introduce to you Dr. Ken Kaushansky. Ken, the floor is yours.
Thank you, Fotis. Thank you for that incredibly kind introduction. I'm sure my mother would be very proud. Today, we have 3 speakers for you. I will be speaking about the basic biology of how the immune system works and how we capitalize on that in the development of vaccines. Dr. Bettina, our Chief of Infectious Diseases in the School of Medicine, will then go on to discuss the clinical trials around 6 of the vaccines, including the 2 that are under consideration for emergency use authorization. And then Dr. Sharon Nachman, the Chief of Pediatric Infectious Diseases, will go on to speak about a lot of the logistics about how the NIH, how the FDA, how the Centers for Disease Control are managing the rollout of these vaccines. So vaccination story begins in 1796 with Edward Jenner. At that time, about 500 million people per century were dying of the disease smallpox. And this photograph, this image depicts Jenner beginning to immunize a child with material from an individual who had cowpox. And because cowpox and smallpox are immunologically related, that actually led to immunization. So much so that as vaccines advanced over the next couple of centuries, smallpox was actually eradicated from the planet in 1977. So to begin, a few terms. Immunity. Immunity is the protection from an infectious disease. If you're immune to a disease, you can be exposed to it without becoming infected. A vaccine is a product that stimulates a person's immune system to produce immunity to a specific disease, protecting that person from that disease. Vaccines are usually administered through injection, but can also be administered by mouth or sprayed into the nose. Immunization is the process by which a person becomes protected against a disease, usually through vaccination, although one could apply that term to acquiring immunity through natural infection, or as you may have heard, protective antibodies were given to many people, including President Trump, that gave a partial protection against the disease. The only problem with that pathway, though, is it's very temporary. The antibodies disappear in about 3 weeks, and hence your immunity, quote unquote, disappears with them. The term herd immunity was also used. And the idea is that once enough people are immune to a disease, the opportunities for a major outbreak of it become so low because, and hence there are people who aren't even vaccinated who become protected in a way. And essentially the bacteria or the virus simply doesn't have enough eligible hosts anymore to establish a foothold, and the infection will eventually die out. So who are the cellular and the molecular players in an immune response? We break immunity down into 2 phases. The first phase is almost immediate, but it's not as protective as the second phase, which usually takes anywhere from 4 to 5 days to really get going. So phase 1, the innate immune system, is composed of cells called macrophages and dendritic cells. They're our first responders. They live at virtually every portal of entry for a microorganism. They non-specifically engulf the invading virus or the bacteria or the fungus, process it into smaller fragments, which immunologists call antigens, presenting those fragments or antigens to more specific but more potent adaptive immune system. Part of this process is a robust inflammatory reaction mediated by molecules that non-specifically inhibit viral replication, molecules that are termed interferons. When someone develops influenza or other systemic infection, the reason we fail so badly is because of the interferon circulating in our bodies, but it's that interferon that's keeping the virus in check until the innate immune system can become activated and effective. So the adaptive immune system is composed of lymphocytes. B lymphocytes produce antibodies, which bind tightly to highly specific sites on the invading virus or bacteria, so to speak, lighting it up, driving targeted destruction by additional macrophages, which recognize the antibody on the surface of the virus or the bacteria. Any single B cell makes only a single antibody, hence the term monoclonal antibodies. There are T cells, 2 basic flavors, T helper cells and T killer cells. T helper lymphocytes are needed to instruct the B cell how to make an antibody, or to make an antibody against what. And they also can help instruct killer T cells to identify and kill specific targets. I'll tell you about those in a moment. The T killer cells then bind to specific parts of the invader and effect several ways to kill the target invader. So with this cartoon, I'll show a virus, the viral antigen, if you will, being engulfed by a macrophage, here labeled APC, or antigen presenting cell. The fragments are chewed up, as I said, and then are presented on the surface of the antigen presenting cell. And in that way, they can interact with T lymphocytes. And so if they interact with a T helper lymphocyte, this lymphocyte now can begin to affect a robust innate immune response. So for example, that T helper cell can instruct the cytotoxic T lymphocyte, CTL, to secrete things like interferons and perforins and granzymes that will then destroy the host that has that virus in it. The T helper cells will also instruct the B lymphocytes to start making antibodies against the invading microorganism. And critical to this whole system is the development of memory. There are memory T cells and there are memory B cells. This is why when you've been immunized or when you have the natural infection, the body is so quick to respond. It no longer takes 5 or 6 or 7 days to mount a response. Because of those memory cells, it takes less than a day to mount an effective response, which is why we don't get sick when we see that same organism a second time. Now, the memory cells need to last a long time in order for us to avoid having to be repeatedly immunized. Okay, so how do we choose the right target for a vaccine? Often it's by trial and error, although sometimes we get some clues and have a logical starting point. And that was very much the case with the rapid production of vaccines against SARS-CoV-2, the pathogen that causes COVID-19. So information gained from studies from 2 prior very pathogenic coronaviruses, the coronavirus, which we now call SARS-CoV-1, that caused SARS, severe acute respiratory syndrome in 2003, and MERS, the Middle East respiratory syndrome, which broke out in 2011, the spike protein of those 2 coronaviruses was identified as an antigenic target for the development of immunity against those infections. And hence, since there is a spike protein in SARS-CoV-2, it was surmised that the spike protein of the current virus would make a good target for a vaccine. A number of studies with the purified virus, blood from patients who recovered from COVID-19, demonstrated that antibodies formed against the spike protein of the COVID agent, the SARS-CoV-2 virus, and those antibodies developed especially at the site where the virus binds to the cell to gain entry and start the infectious process. And we know that those antibodies that appear naturally in people with COVID-19 blocked entry of the virus into the cell. So we had a target, and most of you have seen the illustration on the left, which is the colorized electron micrograph of the virus. The red decorations are the spike proteins on the surface of the virus, and a 3-dimensional structure of that has now been solved. And we know that that green area near the top is where the spike protein binds to the cells in the respiratory tract. And so that is where we want to develop antibodies against, or T cells against, in order to protect against invasion of this virus. So I'm going to tell you about the basic biology of 3 different kinds of vaccines, and then Bettina will expand upon that to talk about what has actually been tested now. So RNA vaccines, this is actually the newest kid on the block. Most vaccines that we have now are because of a protein, for example, the spike protein that's produced and is used as the vaccine. But RNA is the substance that encodes, that tells a cell to make a protein. And so the way these things work is a chunk of DNA called a plasmid. These are replicating DNA fragments. You put the spike protein gene into the plasmid, and you end up with a plasmid that will encode the spike protein. Then in the laboratory, this is turned into a messenger RNA, which can now encode the spike protein. You now inject a nanoparticle encapsulated piece of that RNA. You actually use that RNA as the vaccine. It's injected into the individual. The nanoparticle is taken up by the cell. The RNA is released, and it now encodes and drives production of the spike protein, and the spike protein in your muscle cell will start the vaccination process. A second type of vaccine is based on a viral vector. That is to say, using a relatively harmless virus to carry that spike protein gene into the cell. So the process here is you use a relatively harmless cold virus called adenovirus. You put the spike gene into the adenovirus, you produce the adenovirus, and you use that as the vaccine. Now, the adenovirus has been what's called attenuated. It can't replicate, so it's not going to take over anything. It's very, very safe. The virus carries the gene into the cell. The mRNA is made. It is produced into spike protein, and the spike protein now begins the vaccination, the immunization process. And then the final type of vaccine is called a protein-based vaccine. This is what we use for a lot of vaccines. Here we go back to that plasmid and spike gene, but instead of actually using that to make mRNA, we actually, in the culture, in the test tube, we actually use that to make the protein. So protein is produced, spike protein is produced in the laboratory. The spike protein is mixed with what's called an adjuvant, and I'll tell you what those are in a half a second. Then this is injected into you and I. We now have the spike protein with the adjuvant in us, and that leads to the immune response, both the antibody response and the T cell response. So the adjuvants work, they're substances that are packaged with the virus, with the immunogen, that enhance the immune response. Their primary mechanism is to enhance the persistence of the spike protein at the site of injection, giving a better immune response. They also now attract macrophages, which present that antigen to T cells and enhance the adaptive immune response. And we also think that they probably have a secondary role to craft the type of immune response. But all of the protein-based vaccines are using an adjuvant. So Fotis teed up the notion of how quickly vaccine development has occurred in response to the COVID-19 pandemic. This is comes from a paper that was published in the New England Journal of Medicine a few months ago that talks about the usual serial series development of a vaccine, where we first figure out what a good target might be. We get small scale clinical trial material. We do a safety study in phase 1. We do a somewhat efficacious study in phase 2A. We now begin to scale up production of that vaccine. And now we do a bigger study called a phase 3 study, where we're really looking at safety and efficacy. And then ultimately the FDA might or might not license that vaccine, and then the large scale manufacturing of the vaccine. And this, you can imagine, could take several years to create a new vaccine. Rather, what has happened is we did almost all of this simultaneously. And as a result, the time frame has been squeezed down. Now, please know that the safety element was never sacrificed. The reason this is so much shorter is not because anybody cut corners on safety. They compressed it by doing everything simultaneously. And that is why we were able to get to where we are today much more quickly than it has been in the past. So 38 different vaccine strategies started this process. We're now down to 6 or 7. None have been approved, but we anticipate emergency use approval very soon. And then the final thing I'll leave you with is vaccines don't save lives, vaccinations do, with the implication that a vaccine on a shelf doesn't protect anybody. We need to get vaccinated. Okay, let me introduce to you Dr. Bettina Fries. Bettina is professor of medicine here in the Renaissance School of Medicine and chief of the Division of Infectious Diseases in the Department of Medicine. Bettina is a nationally recognized physician scientist in the field of microbiology and infectious diseases, who joined Stony Brook in August of 2014 from the Albert Einstein School of Medicine. She also has an appointment in the Department of Microbiology and Immunology. She is the quintessential academic physician, the triple threat. She is a clinician, she is a teacher, and she is a researcher. She continues to work on the pathogenesis of chronic fungal infections, working towards developing monoclonal antibodies to help us fight off these devastating chronic fungal infections. A year or so ago, she was elected to be a member of the very prestigious American Academy of Microbiology, and she is now chosen as a distinguished lecturer from the Association of the American Academy of Microbiology. So Bettina has numerous funding since something like 1981. She has published 90 original scientific papers, and she is an expert on vaccines and how vaccines are tested. So Bettina, if you will take it away, we'll learn about the vaccine in clinical trials.
Okay, thank you very much for this kind introduction. Welcome everybody. I'm going to spend my time on giving you some details about the vaccines that are now in advanced clinical trials. And as Dean Kaushansky has already alluded, these vaccines fall into three groups. The first one are the RNA vaccines, the second ones are the viral vector-based vaccines, and the third ones are the protein subunits. The first two groups, so the RNA vaccines and the viral vector vaccines, the difference here is that the person that's vaccinated actually makes the spike protein. And in the third group, you give the person that you vaccinate the spike protein. All three groups of vaccines have the same goal: to generate an immune response that is composed predominantly of neutralizing antibodies that recognize the spike protein and prevent the spike protein from binding to our cells and entering our cells. The other thing that you should know is that this spike protein is huge and big and complicated and needs to fold in a very specific way. And that's why a lot of times people refer to pre-fusion and post-fusion conformations. That basically means that the shape of the spike protein before it fused is a little bit different than the shape of the protein after it fused. Next slide. So the first vaccine we're going to talk about is the Moderna vaccine. This vaccine is an RNA vaccine. The RNA has a name. It's called messenger RNA 1273. And it's a nucleoside modified RNA. That basically means it's a chemically mildly modified RNA, and it encodes this pre-fusion SARS spike protein. And this pre-fusion SARS spike protein is often referred to as S2P. And that basically means that they change the sequence in a way that there are two amino acid substitutions at position 986 and 987. And by changing these two amino acids, you make the protein fold in a specific way. And that way is the pre-fusion conformation. And that is very important because if you have a misfolded protein, you make an antibody response that is not protective. Next. The vaccine is delivered in a lipid nanoparticle. That's basically just a lipid drop. And phase 1 trials have determined that 100 microgram is the right dose. And they are given twice, 28 days apart. The beauty of this vaccine is that it only requires a minus 20 degrees storage. And that makes it different from the Pfizer vaccine. Phase 1 trials have been completed, and they showed that the people that were vaccinated made neutralizing antibodies that also worked neutralizes both pseudovirus and also real virus. Those experiments need to be done under BSL-3 conditions, so in very safe laboratory conditions. And they also showed that these vaccines elicit a CD4 T cell response. That is a T cell response that we think helps the B cell that is stimulated to eventually become a memory cell. So the thinking is when you have a T cell help, that you will have a longer lasting B cell response. So this bias towards Th1 cytokines is preferable. Next. They also reported some CD8 T cell responses in these initial vaccine trials. Next. In the meantime, they have also completed the phase 3 clinical trial that was started in July, and that recruited 30,000 people. And except for some safety data, which still has to be put together, everything is analyzed, and some press releases have already been reported. And they include that this vaccine has a 94% efficacy and 100% efficacy with respect to preventing severe disease. That was reported November 30th. They also reported about 2% of people that were vaccinated had severe side effects. And these are defined as those that prevent daily activity. So that would be fever, muscle aches, headaches, and nausea. And less than 2% of the recipients developed high fever. High fever is like 39 to 40 degrees. That's about 103 Fahrenheit. And they also reported that the reactogenicity, so the soreness and the fevers usually are more pronounced after the second dose and happen within 12 hours. Long-term side effects of these messenger RNA vaccines and all of this spike protein-based vaccines remain theoretical. What we are concerned is that there could be sort of a autoimmune antibody induced, but we don't have any evidence for this. The next RNA vaccine is the BioNTech Pfizer vaccine. They named their messenger RNA 162b2. Again, that's a nucleoside modified messenger RNA, very similar to the Moderna vaccine. Also encodes for a slightly modified spike protein with these two protein substitutions, and is also delivered in a lipid nanoparticle. Also requires 2 doses. They inject 30 micrograms, and the doses are given 21 days apart. And what's different with this vaccine is that it requires minus 80 degrees storage. Once you thaw the vaccine, you actually have to use it within a few days. In their phase 1, phase 2 study, they also showed, they also showed local reactions and systemic events were dose dependent, and they showed that the neutralizing antibody titers were between 2 to 4.6 fold increased, and they showed that they were very comparable to the titers in convalescent human serum. And in their phase 3 trial, which was also started in July, they recruited initially 30,000. They have in September expanded this to include more patients, especially children. And they reported their results on November 18th and reported a vaccine efficacy rate of 95%. They showed that, they showed that of the 170 cases among these 30,000 vaccinated people, 162 cases were observed in patients that had received the placebo vaccine, whereas 8 cases were observed in patients that had received the real vaccine. But none of the patients that had received the real vaccine developed severe disease. And similar to the Moderna vaccine, they showed that they had efficacy consistent across age, gender, race, and ethnicity. We haven't seen the data yet. It's not published, but that's what's reported in the press releases. The 10 severe cases, the majority of them were in the placebo group, and only 1 was in the BNT162b group. And to date, there were no serious safety concerns related to the vaccine reported, and they have followed out people for 2 months now. The Moderna vaccine is going to be presented to the FDA on December 17th, and the BioNTech Pfizer vaccine is going to be reported to the presented to the FDA on December 10th. So that could be potentially licensed next week. Next. Now we are going to move into the adenovirus-based vaccines. There are 2 of these vaccines are in advanced clinical trials. One is the AstraZeneca vaccine. That is the vaccine that was initially pioneered by Oxford University. This vaccine uses a chimpanzee-based vaccine. So the idea is that you need to get this messenger RNA into the cell in order for the cell to produce the spike protein. And you have basically 2 ways of doing it. You either use this nanoparticle, or you can also use an adenovirus. And in the AstraZeneca vaccine, they use this chimpanzee-based adenovirus. This adenovirus has been pioneered in other vaccines, including vaccines against MERS and malaria. And the idea is that because it's an adenovirus that infects chimpanzees and usually not humans, we don't have pre-existing immunity. So, and that is good, because one of the concerns with these adenovirus-based vaccines is that you have pre-existing immunity and you clear the virus, and then the vaccine can never really be efficacious. Again, they have a nucleoside modified messenger RNA encoded into it. They have also a full-length structural spike protein, and that also has some modifications so it folds correctly. The nice thing about the AstraZeneca vaccine is that it can be stored at refrigerator temperatures for up to 6 months, and that positions this vaccine to be used globally in countries that have bigger problems to have a real cold supply chain. You also need 2 doses, and they're given 28 days apart. The phase 1, phase 2 trial showed neutralizing antibodies across all ages, also showed that this vaccine elicits a T-cell response measured at day 14. They also reported injection site pain. You have to realize when you use these adenoviruses, these are all replication incompetent in humans, so they cannot multiply in humans. So you actually inject the humans, you vaccinate them with a large amount of adenovirus, something like 10 to the 10th, so a really high number. And that gives you a soreness in the muscle when you inject within the first 48 hours. The phase 3 US trial was started in August, but actually didn't start in August, because the one that was in the UK was held initially because there were some questions about a patient that developed a transverse myelitis and another patient developed some other neurological symptoms. So they initially held it in the UK for a couple of days, and they started it a little bit delayed in the US. It's also being pioneered in Brazil. So this vaccine is tested globally. Next. AstraZeneca held the trial initially, but then we started it in the US. It started now too. They have on November 23rd reported out their first results, and they said that the vaccine is about 70% effective, which some people felt like, well, it's not as good as the other ones, which is true. But when they looked at the data a little bit more carefully, they found that one dosing regimen actually showed that the vaccine efficacy can be 90%, and that is when the first dose that is given is half dose followed by a full dose. The vaccine regimen, which involved 2 full dose, exhibited a lower efficacy of 62%, and that's why the overall efficacy is reported at 70%. But with the correct dosing, this vaccine should also be a highly efficacious vaccine. Again, 2 doses have to be given at least 1 month apart. No serious safety events related to the vaccine have been confirmed. The 2 that held the vaccine trial were clarified, and the vaccine was proceeded, but we haven't seen the published data. And the next adeno-based vaccine is the vaccine produced by Johnson & Johnson. That vaccine is called NJ78436735. It is basically uses adenovirus 26. Adenovirus 26 is a virus that can cause disease in humans, but rarely causes disease in humans. And it's very similar to adenovirus 5, which is a common adenovirus used in vaccine constructs. It's again a non-replicating adenovirus. It has been pioneered already in the Ebola vaccine and has also been used in other vaccines. They again have encoded a stabilized pre-fusion spike protein into that construct, and they report doses of 5 times 10 to the 10 and 10 to the 11 viral particles, so that shows you the amount of viral particles you inject is really high. The nice thing about this vaccine, it's a single dose. So, and that makes this a really great contender for using it globally if it works and if production can be assured, because you don't have to bring the patient back. So people are very excited about learning how this vaccine is going to perform. Again, it's stable at minus 20 degrees for 2 years, and it's stable at refrigerator temperatures for 3 months. In phase 1, they showed seroconversion in 99% of the participants and also in older ones. And they showed that they had neutralizing antibodies against the virus 29 days post-vaccination, and with high titers in over 80% of the participants. Phase 3 trials, and it also elicits a strong T-cell response, and the immune responses were very similar across all adults. The most local adverse effect was, again, irritation at the injection site and some fatigue and some headaches. And they also, these are just small numbers, but they were mostly resolved within 1 or 2 days. And phase 3 started September 23rd and will include 60,000 participants in the US and in other countries. So now we're going to move on to the subunit, protein subunit vaccines. The Novavax vaccine is a recombinant nanoparticle vaccine, which is also constructed from a full spike protein. So this includes the entire spike protein. Again, these Novavax vaccines, you don't inject RNA, but you inject actually the protein itself. And it can be quite challenging to make protein in such large amounts, in such purity. But the company has figured it out, and they have a proprietary system which involves a cell line that is created in butterflies. And this insect cell expression system is their way of making large amounts of purely and correctly folded protein. And their protein has a little bit of different mutation, and this mutation is in the cleavage site of where the protein is cleaved in 2 parts. And it also has proline substitution, so it has this mutation in addition to the proline substitution. And that just makes sure that this protein is actually expressed in the right shape and in the right conformation. They, in addition to the protein, inject the patient with an adjuvant, which is a adjuvant that has been pioneered by the company, and it's based on saponin-based adjuvant. That's basically a soap-like adjuvant, and that has been used in other vaccines already. And this adjuvant stimulates the immune system, and the thinking is that especially in elderly patients, it will elicit a strong and efficacious immune response. The vaccine and the adjuvant can both be stored at refrigerator temperatures, which is great, but you still need 2 doses, and they're given 21 days apart. And phase 1 and phase 2 trials showed that the addition of the adjuvant resulted in enhanced immune response and allowed us to give less protein and induced a strong T helper response. That's the T cell response, which helps us move the B cells into long-term memory cells. And so in the trial, they give 5 micrograms of adjuvant regimen, gave 5 micrograms of adjuvant regimen, and that induced good neutralizing antibodies. And the antibody titers that they showed were higher than those detected in COVID patients. No serious adverse events were noted, and the side effects were absent or mild in the majority of patients, and again happened usually early on and lasted less than 2 days. Phase 3 trials have started September 25th. They are using 5 micrograms of the spike protein and 50 micrograms of this matrix M1 adjuvant. And they are including patients, at least in the UK, up to 84 years. So they're really trying to do these trials in patients that include also elderly patients, which are at the highest risk. The last vaccine I'm going to talk about is the Sanofi vaccine. They partnered with GSK. GSK has a proprietary adjuvant technology, so they make adjuvants, that's their business. And they got together, and Sanofi also has a butterfly-based expression system to make spike protein, and they have also their specific spike protein, and it's combined together with this GSK proprietary adjuvant technology. They haven't really released that much data on it, but they have immunized individuals, and they have shown that that these patients elicit neutralizing antibodies, and they are and we are anticipating further results in early December. And now I will introduce Dr. Nachman. And Dr. Nachman is my colleague and is currently a professor of pediatrics, the associate dean of research and director of clinical trial office. She's also the division chief of pediatric ID. She has over 150 publications, many of them in top journals like New England Journal of Medicine. She has been NIH funded for almost 20 years, and the stage is hers.
Thank you very much, Bettina. So I guess the key questions that I have for this audience are, what are the next steps? What is NIH doing to combat COVID? Well, NIH has operation, and I'll go through all the questions first. What is the relationship between the FDA, the ACIP, or the Advisory Council for Immunization Practices, and the states? And the last key question is, what about rollout and tracking? How are they going to happen? So what has the NIH been doing? Well, they have something called Operation Warp Speed, and it's broken out into several different categories. Who participates in Operation Warp Speed? Well, there's the vaccine treatment evaluation units, the HIV vaccine networks, the HIV treatment networks, and the pediatric maternal HIV networks. All of us together combined serve as both the sites and the membership for this Operation Warp Speed, with the idea that having a critical mass of investigators and a critical mass of sites will allow us to ask the right science questions, develop the protocols, roll them out in a timely fashion, and get the answers for everyone. So the co-VPN, their job is to look at vaccines and prevention. The ACT UP group is idea to look at treatment. Next. The treatment studies then fall into several categories. Do we want to treat somebody who actually has COVID? Do we want to treat someone who's been exposed to someone with COVID? Or do we want to treat the household around someone who has COVID? Those treatment studies are underway, some of which are being done here at Stony Brook, and we are doing a treatment study right now with monoclonals, with the idea that as new drugs roll into the pipeline for treatment of COVID, both as outpatient as well as inpatient, our site, like many others, in Operation Warp Speed, will be able to use those treatments locally. With regard to some of the prevention studies, we've also been looking at monoclonal as well as remdesivir for special populations. And Stony Brook is a site for using remdesivir in pregnant women, recognizing that the licensure, while approved for that group, actually doesn't include any data on that group. So Stony Brook is a site for using remdesivir for hospitalized women who are pregnant with COVID. With regard to the vaccine studies, they fall under several categories. We have to think about vaccinating adults, children, and pregnant women. All of the studies that you've heard Bettina talk about usually started at age 18. Some of them included a very small and limited population of age 12 to 18. Were those populations of 12 to 18 enough to get licensure? The answer is unfortunately no. And every company that did include very small populations of that age group is now instructed by the FDA to go back and try it again and target 12 to 18-year-olds as well as children. In fact, we are now looking at bringing several of these studies for 12 to 18-year-olds, and in fact, down as low as age 2 to as high as age 18 to Stony Brook. We can't, however, forget pregnant women, recognizing that each one of the studies that Dr. Fries discussed actually excluded not only pregnant women, but adults who had cancer, adults who are on immune suppressives, and a whole host of other patients. So if your patient or you are one of those populations, we actually will have no data on how effective that vaccine will be. And part of the FDA and the ACIP group's discussions will be which populations were included in the vaccine. So can we say that the data applies to them versus we don't have a clue, we're going to have to restudy them. Pregnant women, though, are targeted for the upcoming studies for pretty much all of the vaccine companies that Dr. Fries discussed. And we assume or hope that those vaccine studies will be rolling out first quarter of 2021. Next slide, please. Well, what about the next steps? What do we have to do in order to get to the FDA? The first thing is that each company has to have all of their data reviewed by what we call a DSMB, or the Data Safety Monitoring Board. What's unique about this situation is that the exact same DSMB is reviewing across all of the pharma companies, all of their data. Why is that important? Because there may be a small signal of safety per each vaccine study. And only having the same DSMB will allow us to look for that vaccine safety signal across studies. So is it possible that we've seen it in both mRNA vaccines? Is it possible that we've seen it in both adenoviral vector vaccines or both adjuvant-driven vaccines? The DSMB will have enough data because they're looking across studies, not only at individual studies, to say, is there or is there not? So instead of having a sample of 30,000, they actually will have a sample of 60,000+ to look for those rare or infrequent safety events. Now that the DSMB has reviewed it and said, yes, go ahead, the next step is to go to the FDA. The FDA has pulled together a totally independent advisory panel consisting of 15 voting members and about 20 non-voting members. Amongst the voting members, they include community reps, which means that it's not just up to the scientists to discuss it, it's up to community to say, what does this mean to me? How are we going to understand the data? And who does this vaccine apply to? Now that the FDA independently reviews it, they make a recommendation to the FDA, and the FDA may say, yes, you have enough for EUA approval. But even after the FDA says that, next, it has to go to another group. EUA approval, remember, is not licensure. And as we've all seen in the start of the pandemic, we've had a lot of tests that were EUA approved that then fell out of favor. So it is not enough for you to get your EUA approval. You have to complete all the information needed for licensure in order to keep going. The EUA approval will be good for a while. We will be able to roll out vaccines, but if we want to use these vaccines past the short term of getting them out, we're going to have to get all of the licensure information. What's included in licensure is going to be not just the short-term safety data, but all of the long-term safety data. And we're going to discuss in a minute how to get that long-term safety data. Next slide. After we get the EUA approval, then the Advisory Council on Immunization Practice, or ACIP, then takes that exact same information and says, while you have some kind of approval, who do we think should actually be getting this vaccine? And because of that, they've actually broken it out into a whole bunch of different groups. Next slide. Who goes first? Well, the group 1A, according to the ACIP, should be healthcare personnel, nursing home staff, and patients. What's the problem? The problem is that's 21 million people. Next. In cohort 1B, they've included essential workers. We have 87 million of those. High-risk medical conditions, we've got about 100,000 of those. And then if we just take plain old adults over 65 years of age, we've got another 53 million of those. So just looking at cohort 1B, and there's some overlap between cohort 1B and then cohort 1C, which is probably the adults over 65, we need to think about how are we going to get enough vaccine for everyone to get their required 2 doses, except for the Janssen, which needs 1 dose. Everybody else needs 2 doses. Is it better to give everybody 1 dose and nobody 2 doses? Or is it better to give some of the people 2 doses and the other guys wait? Right now, the recommendation from every single advisory group has been to give those that can 2 doses, and those that we can't assure getting 2 doses, holding on for the next week or 2 weeks or 3 weeks to make sure we have enough supply so that they can get their own 2 doses. Remembering, you cannot mix and match. You can't get one messenger RNA vaccine from Pfizer and then dose 2, same messenger RNA vaccine type from Moderna. You're not allowed to do that. So if you can't get dose 1, you shouldn't, rather dose 2, you shouldn't be getting dose 1. When I think about essential workers, go back for a sec. When I think about essential workers, the laundry list is enormous. Food and education, energy, police, firefighters, manufacturing, communication, agriculture, food service, transportation. It's pretty much everybody that you know in your life. So in order to make sure that we have enough to give those people 2 doses, we really have to hold out and count how many doses do we have in our hands right now to make sure that we're going to be able to give them their 2nd dose. Next slide, please. Now, what about rollout? As you heard from Dr. Fries, each vaccine has different requirements for the cold chain. Is it come in a vial that it's multi-dose or single dose? How are we going to administer it? Who's going to track it? All of those needs are reflected in each state, had to develop a plan exactly where it's going to store it, how it's going to get from place A to place B, how it's going to be transported there, who's going to check it in, who's going to check the logs, and then who in fact will actually be administering that dose. Next. We have to prioritize within groups as more vaccine becomes available because, as I've said, you can't mix and match the doses. Next. We have to reflect on the data from each study as to how representative of the population. For example, children in nursing homes are excluded from every study. From the adult studies, they're excluded from all the pediatric studies, yet they are a real population that we see. Not only the kids that are in nursing homes, but kids that have chronic medical conditions. We can't give any of them vaccines, and no company wants to study them until we've given all the other populations who are well their vaccine. Why is that sort of silly? Because in fact, those are the kids who are going to get sick and wind up in the hospital. Next slide. So we ask ourselves, are you looking like the recipients of the vaccine in that study? Next slide, please. So what about tracking? And tracking is going to be very important. There's the New York State Immunization Information System, and I put the link on over here, which is available to every provider. As per Dr. Zucker from New York State, this tracking system is going to be required for everyone to be enrolled on to see which vaccine they got. So if you go to one place and get a vaccine and it's time to get dose 2, and you don't know what you got or who it is, anyone who's going to be giving you dose 2 can open this tracking system up, see what you got, when you got it, so they can be assured to make sure that you get the exact same manufacturer for dose number 2. So it's going to be very, very important for us to continue to track what you got. In addition, this tracking system allows you to report adverse events. So if you got dose 1 and you had an adverse event and shouldn't be getting dose 2, your physician or anybody who is in the medical profession will be allowed to go on this information system and write down what your adverse event. This information can all be printed up as well, so that if you need documentation, like you're flying on an airplane or going for a trip, to say, yes, I actually got 2 doses of vaccine A or vaccine B, this information will be available to be downloaded from the information system. So what are the next steps for us? Well, number 1, we still have to stay safe. We have to continue to wear masks and social distance. And also, we want people to not be afraid of going to the doctor. We want them to keep up with all their current medical needs, because it's not going to be helpful if you're sick when we want to vaccinate you. I see there are a lot of questions in the chat box, and I'm going to leave them to be directed by, I think, Dean Kaushansky. You're going to ask the questions and moderate the next session.
Actually, I think that Fotis is going to be asking the questions because I couldn't see them as they were rolling in.
So I would love to, but because we are running late and there is many questions and I have a meeting that starts in 5 minutes, I have asked Nadine to do the questions so that we can run over, okay? Thank you all for great presentations.
Okay, one of the questions is, if once vaccinated, can he or she spread the virus?
So I can take that one. And the answer is no, you cannot. If you're vaccinated and you've made an immune response, you should not be, first of all, none of these vaccines are live, so they will not give you infection, but you should not be able to spread virus because you should not be infected. pretty much every one of the studies collected antibody testing before they were vaccinated and are looking at the change in antibody over time. They're not just collecting nasal swabs to see if you were infected or to see if you're symptomatic, but also to look at the change in antibody. So that information will help solidify the answer. But at this point, no one is expecting that if you are exposed to infection, that you will be able to pass it if you've made antibody.
How long is the immunity expected to last?
That's not known yet. So we know that from natural COVID infection that we have seen patients that have gotten reinfected. We know that the majority of patients that get natural COVID infection make antibodies. The majority of them still have antibodies 4 or 5 months out. But we don't know whether this vaccine has to be boosted again a year from now, whether these antibodies are going to last longer. What we know is that the vaccine regimens are engineered in a way that they give you both an antibody response and a T cell response, thinking that giving you good T cell response and the right T cell response is going to allow you to make a longer lasting B cell response. We have vaccines that last an entire life. I mean, you know, most people get 2 or 3 measles shots in childhood and they never get a measles shot again. And so we are hoping that this will last, but you know, it may be that if this virus persists in our community, that we need to get re-immunized.
Another question here is, how does the consumer decide what vaccine to take, but also in regards to insurance? You know, how do they choose?
So ACIP, once they approve a vaccine for a population, automatically all the insurances will then pay for that vaccine. There's not going to be allowed to any insurance company to say if ACI approved it for that population that they are not going to pay. So that's first. The second is that pretty much all of the vaccine manufacturers have also agreed to provide the vaccines for free at this time. So it is not expected that there will be a bill generated from the vaccine. There may be a bill generated from an administration, but certainly not from the vaccine at this time.
And lastly, right now, we're looking at having 2 vaccines licensed in December, hopefully. And the AstraZeneca vaccine is probably also going to come up for license fairly fast in the US. The other ones are not there yet. So, you know, I'm sure Dean Kaushansky is involved in the discussion, how many vaccines are we going to get at Stony Brook, who is going to get them. You know, right now, we don't have enough vaccine for everybody. And so the question is not going to be so much, which vaccine am I going to choose, but more, am I going to be able to get a vaccination? That's our primary concern now, is to get vaccination.
And in fact, just yesterday, I was on a conference call with the chancellor making the very argument that we are an ideal site to store vaccine. We have actually a storage capacity for about 4 million doses of vaccine in the MART building and in the hospital pharmacy, even at minus 80 degrees. And so we have a plan. A lot of people here at Stony Brook have put together a very, very good plan to immunize our healthcare professionals, our people at risk, the Long Island State Veterans Home. And then when it comes time to immunizing large populations, where we're going to be doing the immunizations is actually quite close to the outside of the Health Sciences Center. And we can even go and start immunizing in that way. So we are well prepared. Another question that I can now see the questions. Another question that came up is, are people going to get to choose? We hope they're probably not going to get to choose. It's going to be what's available. And since the efficacy of at least the 3 major phase 3 trials that have been reported are all roughly the same, in the middle 90% efficacy, which, by the way, nobody said it, but that's incredibly high for a vaccine. And so I think we lucked out, or incredible, well-designed vaccines across several different types of vaccines. So it probably won't be critical which vaccine is available for you. But as was mentioned, The 2 RNA viruses are probably going to be approved first for use, widespread use in people, for use in healthcare professionals, and we hope very soon be able to administer it. We here at Stony Brook are ready.
There's another question here that says, is there a point after being vaccinated that we are able to stop wearing masks?
So everyone is trying to model that right now. How much of a population do we need to get masks? What's the critical new infection rate in that population as well, in order to be able to say, no, we don't need masks? Most of the people that we have talked to have suggested that after we have the adults vaccinated and start getting into the pediatric vaccines, hopefully by the summertime or early fall, that's when we will be able to say we don't need masks anymore. But that really depends on people refusing vaccine or not taking vaccine or not getting their second dose or not being careful until that time.
But the bottom line was on Sharon's last slide. We have to be very, very careful. We have to continue, and vaccines don't save lives, vaccination does. And so as soon as I'm eligible to get a vaccine, I'm getting a vaccine. I suspect Sharon and Bettina are in the same boat, and we would encourage people to become vaccinated.
Okay, so I'm going to have this as the last question unless Dr. Kaushansky wants to add anything or read any more. One of the questions here is, if someone recovered from severe COVID-19, does he or she need to be vaccinated?
So some of the vaccine trials, actually, the vaccine trials didn't differentiate whether you had COVID before or not. And so the answer is that they sort of anticipated when they designed the vaccine that they wouldn't be able to tell whether the patients already had it or didn't have it. And I think that's how we're also going to proceed with vaccination, because if you had really severe COVID and you were really, really sick, maybe you didn't make an antibody.
The other part to that and it's a great question. One of the other parts to that question is the question of, is the natural infection as good at stimulating a robust immune response as an engineered vaccine is? As Bettina said, these, the adenovirus vaccines are putting a tremendous amount of antigen into your muscle, which is producing a very large amount of spike protein and consequently a very robust immune response. The protein, as you heard, is being engineered to be maximally immunogenic. And so there are a lot of reasons to think the adjuvant further bolsters one's immune response. So there's a lot of reasons to believe that a vaccine is better at inducing robust immunity than the natural infection. So even though you might have had COVID-19, I hope you haven't, but if you did, it is probably still very much worth your while to become immunized with a vaccine when it's your turn.
So I just wanted to add one thing to that before we close, in that in the rhesus monkey model of COVID, where all these vaccines had to be tested before they got to humans, even for the phase 1 study, exposure to natural infection versus vaccination was actually in fact different in the animal model. And in the animal model, the antibody response to native infection, while it was there, was not nearly as robust, nor did it stay as long as antibody production after vaccination. And that is true for 100% of the vaccines that were discussed this afternoon. So having COVID, in fact, you made an antibody, but it wasn't as high enough and stayed around long enough as you did when you got the vaccines.
So I got the message, and we will we'll end our mini symposium on vaccines. I want to thank Bettina. I want to thank Sharon. I want to thank Fotis for this opportunity for us to share both biological, clinical, and regulatory insights into a very important and very exciting topic. I will just close with why we are working so hard towards a vaccine, because as you know, we're now into round 2 here in New York. And just today, our hospital census of patients with COVID-19 or suspected COVID-19, we're up to 85 patients today. And every day for the last week or so, we've seen 10 more patients in our hospital. So it's out there, it's coming back at us. Masks, social distancing, being safe, staying out of big parties, staying just within your pod is the best way until the vaccines are ready for
And
you.
wash your hands.
And wash your hands. Thank you, Bettina. And that's good for a lot of things.
That's good.
And the masks are good for a lot of things also. Influenza, it's flu season. So thank you all for attending, and have a great, great day, everybody.